Hi everybody, and welcome back to the latest VodCast for Honors Biology at Desert Ridge High School. And we are continuing our scientific mystery of an investigation into what genes are made of. When we left the story off, we were talking about Mr. Thomas Hunt Morgan, who was an early researcher in the early part of the 20th century. And what Thomas Hunt Morgan established was that there are these things called genes, and they reside somewhere on a chromosome. And we know that chromosomes are made of both protein and DNA, and we're trying to figure out what these genes are exactly made of. And so the prime suspect in this investigation right now is protein. Protein is a very complex molecule, which people knew in the early 1900s, and DNA is relatively simple. There are only 4 bases, and they were known to occur in roughly equal proportion. So at this point, we want to look at our various suspects and say, well, what would this suspect look like? What would the profile of the suspect? What would what are we looking for in a genetic material? Well, the thing that everybody agreed on at the time was that whatever this stuff is, whether it's protein or DNA or something else, it had to be something that could store information. It had to be something that could be copied and passed on, which obviously our traits are passed on from one generation to the next, and it had to be inherited according to Mendel's laws. So these were the 3 characteristics that people knew that whatever genes were made of, this is what they would have to look like. Okay, one of the first people that was a researcher in the early 1920s and 1930s was a guy named Frederick Griffith. Mr. Griffith was British, and he actually was not interested in genetics or this. He didn't know that he was even working on this particular mystery. What he was a physician who was working with white mice looking for a cure for pneumonia. There are several different types of pneumonia. One of them is caused by a bacterium. And the discovery that he made accidentally was that bacteria could be transformed. Now, a sort of a sad part of the story is that Mr. Griffith was working on this problem, and during the Blitz in the early days of World War II, the Nazis were bombing London, and Griffith was killed at this time, so he was not able to conclude his experiments, but he did get a good enough start that other people could finish it. Okay, here's a little summary of what Griffith's experiment involved. First of all, you have to understand that there are 2 different strains of this particular bacteria, an R strain and an S strain. The R and S stand for rough and smooth, and this is sort of like eye color in people. So these are both the same species of bacterium, both the R and the S, but they're just a genetic variant or a genetic variation in that same species. So the R strain is harmless. Okay, it's the pneumococcus bacteria. If you inject this into a mouse, the mouse does just fine. However, the S strain of this bacteria, if you ingest that into a mouse, the mouse quickly gets pneumonia and dies. So one of the things that Griffith was trying to determine is it the living bacteria it produces, or is it some chemical that the bacteria produces that causes the pneumonia? So what he did was he took this S strain bacteria, put it in some liquid, boiled it so that the bacteria were dead, and then injected that into the mouse. And what happened? Well, the mouse lived just fine. So what this showed was it was none of the chemicals that the bacteria produced. It was actually the living bacteria themselves that caused the disease in the mice. So the next thing he did, which was rather interesting, was he took again some of these dead boiled bacteria that would otherwise cause disease. So he took those, boiled them, and then injected them along with some living R strain bacteria. Injected those into the mouse, and guess what? The mouse dies. More interestingly enough is that when they sampled the tissue from the mouse, from the dead mouse, they found that there were living S-strain bacteria found in the mouse. Now, I should make clear here that these were not the original bacteria that were changed, but because of the fact that bacteria reproduce very rapidly, these would have been the offspring of these original bacteria that were placed into the mouse. So the question here that I want you to be sure that you understand is, what does this have to do with genes? And the answer to that question is that this trait that makes these bacteria lethal is something that is inherited. It's a trait that is passed on from one generation of the bacteria to the next. So here we have a situation where we have some bacteria, there's some chemical in this that are able to change the offspring of the R bacteria and make them look like the S bacteria. So there is some chemical that is transforming the offspring of these R bacteria. Okay, this is as far as Griffith was able to get with his experiment. And then along came another fellow named Oswald Avery, who was an American working in the east coast of the U.S., was also working for a cure for pneumonia, but was also interested in genetics. So Avery kind of picked up where Griffith left off and was able to begin trying to isolate the particular molecules from those heat-killed bacteria that are going to actually cause the disease. Okay, so in other words, what he was trying to find out is exactly which type of molecule is causing this transformation. So what he did was he again took some of these S-strain bacteria, boiled them so that they were dead, and then he took each type of molecule from that liquid mixture. So the first thing he did was he took the lipids. So we've got the lipids from these dead bacteria. We mix them with some of the living harmless bacteria, inject that into the mouse, and what do you think happened? The mouse does just fine. Next thing we take is the carbohydrates. We take out the carbohydrates from this liquid. We mix those again with the R-strain bacteria, inject those into the mouse. We hold our breath, and the mouse does just fine. So now we've determined, well, it's not the lipids, it's not the carbohydrates. So at this point, we're going to do the proteins, and this is the point where everybody said, okay, it must be the proteins. It's not the lipids or the carbohydrates. It's got to be the proteins that are carrying on that trait from the S-strain bacteria to the R-strain. So we take the proteins, we mix those with the R-strain bacteria, inject those into the mouse. Nothing happens. The mouse is just fine. Well, the experiment was repeated several times because people were convinced it had to be the proteins. It's not the proteins. The mice continually did just fine. Then, just to make sure that they didn't miss something, they took the nucleic acids, which everybody thought were not very important. They took the nucleic acids, which included the DNA out of the dead S-strain bacteria, mixed those with the R-strain bacteria, inject those into the mouse, and what do you know? That was the mouse's last day. So what that showed, well, what do you think that showed? I hope you said that shows that the nucleic acids or the DNA is actually the material that was transforming these otherwise harmless bacteria back into the S-strain bacteria. And that's what the results showed, was that the nucleic acids, the DNA, is the material that was carrying the genes. Okay, now you might think that the case was closed, but you'd be wrong. Scientists are a pretty fussy bunch, and a lot of scientists did not buy that. They said that maybe the DNA was just something that was used to activate the genes and that the genes were really proteins, and they thought this because DNA was just too simple to be very important. So the next thing that people said was, well, if we had some knowledge of the structure, if we knew something more about this molecule, then maybe we could see that how important the DNA is. And so that's where the next group of researchers came in, and one of the first people to come in was this sharply dressed young man who was named Char Gaff. This is what Char Gaff looked back in the 1930s. And at the time when Char Gaff came along, people assumed that the amount of adenine, thymine, cytosine, and guanine were always the same, were always 25%. Other people had looked at this, but they hadn't looked very closely, and most people were satisfied with that. What Char Gaff did was he took took samples from different organisms. So this is Homo sapiens, which is humans, Drosophila melanogaster, which are fruit flies, Zea mays, which is corn, and then the rest of these are all different types of bacteria. He took those and looked very closely, looked very precisely at the amount of A and T and G and C that occurred in the samples. And he found a couple of things. First of all, if you look at this, you see that in humans, the amount of A and T is the same, but it's different than the amount of G and C. And when we look at different organisms, we see a couple patterns emerging. We see, first of all, different species, we have different percentages, right? Here we have 31 and 19, here we have 27 and 22, here we have 25 and 24, so the percentages are different for different species. The other pattern that we see is that the amount of A and the amount of T is always the same, the amount of G and the amount of C is always the same. And so what Char Gaff concluded, these came to be known as Char Gaff's rules, were this, that the amount of A and T and G and C is different depending on which species we're looking at. The other thing that we see is that in each species, the amount of A is equal to the amount of T, and the amount of G is equal to the amount of C. And this is true in every type of organism. Okay, so this turned out to be a very important clue for Watson and Crick. Okay, and if you're wondering how that could possibly be, think of puzzle pieces. Okay, this was a puzzle piece, this was a clue, this was not the answer, but this was certainly an important clue that they were able to use. Another person who was working at the time was a researcher named Rosalind Franklin. Rosalind Franklin, during her lifetime, was not very famous, but in recent years she has become rather more well known. She published an important paper in 1953 at the same time as Watson and Crick. She used a technique called X-ray crystallography, and this is a fancy name, and it essentially boils down to this. If you take an X-ray beam and you shine it through some form of a crystal, in other words, what they did at the time was they took DNA, took enough purified DNA to make a crystal, shone this X-ray light through it, and then it would project an image onto X-ray paper, similar to what you would get at a doctor's office. Of course, the resulting image doesn't look anything like you would see at a doctor's office. It had this sort of ghostly X pattern to it. And what this was used for, or what people found from this was evidence that DNA has a spiral structure, which we call a helical structure. Spiral and helical both mean about the same thing. So this was some evidence that was available to her, as well as to other researchers at the time, that DNA was a spiral form. Well, one of the people who knew Rosalind Franklin at the time, of course, were these two guys, James Watson and Francis Crick, who were also working in London at the time. And what they did was they took a different approach. They basically tried building models. They had some information. They had some information from Rosalind Franklin. They also had some information from Char Gaff as well as others. And they took knowledge and observations from other people and made a model. And what the model essentially consisted of was looking at the bases, looking at these nitrogenous bases, thymine, cytosine, adenine, and guanine, and they made some cardboard cutouts and just kind of started sticking them together to see how they fit. And one of the things that they noticed was that when they built this model, that the G and the C fit together very nicely, the A and the T fit together very nicely. And this, lo and behold, was fit very well with Char Gaff's rules. So now they had a potential structure that not only fulfilled Char Gaff's rules, but also suggested a structure whereby the molecule could copy itself. They published a paper in April of 1953 in Nature magazine, which is a scientific journal, and it contained this statement, which has become sort of a famous statement. They said, "It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material." And of course what they were talking about was the fact that these two strands of DNA could be separated, and each one was a sort of a mirror opposite of the other and could be used as a template to form that particular sequence. So that was the first thing in there, or that was one statement that was in there. There was a lot of other things in there. Another thing that was in there, of course, there's a lot of controversy regarding their relationship with Rosalind Franklin and whether or not credit was given. And I do want to mention this just in fairness to all parties. This is one of the last lines of the paper, and it says, "We have also been stimulated by a knowledge of the general nature of the unpublished experimental results and ideas of Dr. M.H.F. Wilkins and Dr. R.E. Franklin and their co-workers at King's College London." I mention this because a lot of times I meet students who sort of have been misinformed. They say, well, Watson and Crick ripped off Rosalind Franklin. There's certainly a lot of controversy surrounding the discovery, but I think ripped off is a little bit harsh. The sad part about Rosalind Franklin is that she died in the late 1950s. These guys were awarded the Nobel Prize along with Maurice Wilkins, this guy who's also named here. And it's a lot of people feel like had she lived, she would have also shared in that prize. Of course, nothing is certain, but they don't award the Nobel Prize posthumously. So, you know, it is a sad story because she died, and of course we'll never know. But ripped off is a little bit strong. Could they have given her more credit? Well, possibly. But like I say, there's enough controversy, but they're certainly not criminals. Anyway, well, that brings us up to where we can start talking a little bit more about the nature of DNA. And so that's all for this VodCast. This has been a VodCast for Desert Ridge High School in Honors Biology, and I hope you have a great day.